Hello everyone, I am Dr. Suman Bhandary, Assistant Professor, Department of Biological Sciences, School of Life Science and Biotechnology of Adamas University. In my first two lectures of this particular module, isolation of industrially important microbial strains and fermentation media, we have discussed about the sources of industrially important microorganisms. Then we have discussed about the screening strategies. We have discussed about culture collection, their role, and we have discussed about the preservation methods of the isolated industrially important microbial strain. Today, we will be discussing on industrial strain improvement and strategies for strain development. But before entering into the topic, let us discuss about what are the learning objectives of today's lecture. At the end of this lecture, you will learn about the specific trait or specific characteristics that need to improve for a given industrial process. You will understand the genetic basis of desired traits in microorganisms, and also you will learn the methods for introducing genetic variability and selecting strains with improved traits. Let us discuss what are strains. Actually, a microbial strain is a specific variant, a subtype, or genetic type of microorganisms such as bacteria, yeast, or fungi that is distinguishable from other members of the same species based on certain characteristics. And in microbiology, the term strain is used to denote a group of microorganisms with distinct genetic features or phenotypic traits that particularly set them apart within a particular species. And these differences can arise due to genetic mutations, variations in plasmids, or other factors. One of the example of a particular strain, that is E. coli strain K12. Now, we need to address the key points about a microbial strain. Means a microbial strain must include what type of characteristics. So there should be genetic variation, there should be phenotypic traits, there must be nomenclature, means microbial strains are often identified and distinguished by specific designations such as alphanumerical codes or a name. Just I have mentioned Escherichia coli K12, Saccharomyces cerevisiae BY4741. These are the specific strains within their respective species. Now, we will discuss about strain improvement. So, the science and technology of designing, breeding, manipulating, as well as continuously improving the performance of microbial strains for biotechnological applications, for applications in fermentation technology, applications in pharmaceutical sciences is referred to as strain improvement. And the science behind developing improved cultures has been enhanced recently by greater understanding of microbial biochemistry as well as microbial physiology, or coupled with advances in fermentation reactor technology as well as genetic engineering. In addition to that, the availability and application of user-friendly analytical equipment such as high performance liquid chromatography or high pressure liquid chromatography, mass spectrometry that raised the detection limits of metabolites have also played a critical role in screening improved strains. So improving the commercial and uh, technical capability of microbial strains has been practiced for centuries through selective breeding of microorganisms. And in making specialty foods as well as fermented foods like beverages like alcohol, beer, wine, vinegar, yogurt, cheese, etc., etc., or a specific strain of bacteria or fungi isolated by chance have been employed to obtain desirable and palatable characteristics. Now, with integrated knowledge of biochemistry, chemical engineering, as well as physiology, microbiologists have taken a more scientific approach to the identification of microbial strains with the desired traits. Now, let us discuss about the attributes of an improved strain or improved strains. So, microbial strain improvement cannot be defined simply in terms of modifying the strain for overproduction of bioactive compounds. Strain improvement should also be viewed as making the fermentation process most cost effective. So some of the traits unique to fermentation process that make a strain improved should have the ability to assimilate inexpensive as well as complex raw materials efficiently. It can alter product ratios and eliminate impurities or byproducts problematic in downstream processing. And basically, number of byproducts, if a microbial strain produces, it will create hindrance in the downstream process. Means when we are trying to separate the product. Now, third point is that reduce demand on utilities during fermentation. Means some sort of microorganism need air, or it may produce heat during fermentation. So we have to constantly supply cooling water inside the fermenter. So a huge amount of power consuming this chemical processes or the fermentation processes. Sometimes excrete the product to facilitate product recovery, provide cellular morphology in a form of suitable or product separation. Fermentation time should be short. Overproduction of natural products or bioactive molecules not synthesized naturally. As for example, insulin. So, what type of strain we consider as an ideal strain, or what should be the ideal characteristics of a strain? The first thing is genetic stability. Second, rapid growth. Means efficient production of target product. And the route of biosynthesis of this target product should preferably be well characterized. Limited or no need for growth factors like vitamins or amino acids. And they can utilize a wide range of low cost and readily available carbon sources. Amenability to genetic manipulation. They should be non-pathogenic and they will not produce any toxic product or byproduct. Readily harvested from the fermentation tank and if they are producing any intracellular product, readily we can break the cell. So these are the characteristics, and if a particular microbial strain have this kind of characteristics, we can consider that particular strain as an ideal, industrially important microbial strain. Now, let us discuss about the need for strain improvement or the purpose for strain improvement. You know that microbes like bacteria, actinomyces, fungi that live freely in soil or in water and produce novel compounds like antibiotic, like vitamins and organic acids, etc., etc. And these compounds are known as novel compound as because they are of commercial interest. So when isolated from their natural resources, they are not ideal for industrial use most of the cases. Accidentally, Alexander Fleming has discovered the fungi Penicillium notatum, which can produce penicillin, that is an beta-lactam antibiotic. But most of the cases, it has been observed that the naturally occurring microbial strain are not ideal for industrial use. And in general, while strains cannot make the product of commercial interest at a high yield, so we have to make them economically viable. And in nature, metabolism is carefully controlled to avoid wasteful expenditure of energy as well as accumulation of intermediates and enzymes needed for their biosynthesis. So, this tight metabolic as well as genetic regulation and synthesis of biologically active compounds is ultimately controlled by the sequence of genes that is present within the DNA. And to improve these wild type strains of microorganisms, the sequence of these genes that is present in the DNA that program the biological activity, we need to improve. So to improve microbial strains, the sequence of these genes must be altered or must be manipulated. In essence, microbial strain improvement requires alteration as well as reprogramming of the DNA, mostly the genes. And we have to reprogram of the genes in a desired fashion to shift or to bypass the regulatory control as well as the checkpoints. And such DNA alterations enable that particular microbe to devote its metabolic machinery to producing the key biosynthetic enzymes and increasing the yield of the product. In some cases, simple alteration in DNA can lead to structural changes in specific enzyme that may increase its ability to bind to the substrate as well as enhance its catalytic activity or make itself less sensitive to the inhibitory effects of the metabolites. On contrary, when the changes are made in the regulatory region of the gene, mostly the promoter side, it can lead to degradation of the gene expression as an overproduction of metabolite. A typical example is overproduction of the enzyme amylase, where specific constitutive mutants have been developed that produce the enzyme even in the absence of the inducer, that is starch. So we need to address now, so what are the targets of strain improvements? So initially we have discussed the targets briefly. that genetic stability, non-toxic to human, non-pathogenic to human, growth must be rapid, and the metabolite product produced in a biosynthetic approach, the complete protocol, the complete flow of that biosynthesis should be evaluated early. Now, we will discuss about the strategies through which we can improve the microbial strain, or we can develop microbial strain. Now, several procedures are employed to improve microbial strains, and all the procedures basically applied to change the DNA sequence. And these changes are achieved by either by mutation or mutant selection, genetic recombination, or the modern techniques, that is recombinant DNA technology. Or we can say the modern DNA splicing technique, which is known as genetic engineering. So each procedure has distinct advantages, and in some cases, a combination of one or more technique is employed to attain maximum strain improvement. Let us discuss about mutant selection or mutation. Now, microorganisms generation after generation generally inherit characteristics identical to their parents. However, when changes occur in the DNA, they too are passed on to the daughter cells. And that particular change are inherited in future generations. So this permanent alteration of one or more nucleotides at a specific site along the DNA strand is basically known as genetic mutation. The strain that harbors the mutation is basically known as mutant strain. Although a gene consists of hundreds or over thousands of base pairs, a change in just one of these base pairs can have a significant change in the function. And that can reflect in their operation as well as expression of the gene. And expression of the protein, which is basically a product of the gene. Now, a mutant, by definition, will differ from its parent strain in genotype. So, the precise nucleotide sequence in the DNA of the genome, and in addition to this visible changes of the mutant, when it reflects the phenotype of the mutant that is altered from the parent strain. So an example we can give here, the first culture that Alexander Fleming was used, it is Penicillium notatum, and that could grow only under static condition, and it yields low concentration of penicillin. But in 1943, a new strain of Penicillium chrysogenum, was isolated. Penicillium chrysogenum NRL1951, which was further improved through mutation, basically by UV treatment and X-ray treatment. And that results a better yield. And no, you will know that today most of the penicillin producers, they're not using Penicillium notatum that has been used by Alexander Fleming. Rather, they're using this improved strain of Penicillium chrysogenum, and that can grow aerobic starred fermenter. So in continuous starred tank fermenter, aerobically this particular improved strain can grow, and also it gives 55 times higher penicillin yield than the original static culture. So now we can discuss that here there is a flow that how you can basically do this, how you can change this. So here you can see that mutation can be done physically by using X-ray, by using UV ray, or using some gases. Then what we have to do, we have to dilute it, and then we will go for random clone selection. So later on, you can screen this using sophisticated techniques like mass spectrometry or high pressure liquid chromatography that can be used for secondary screening, whether mutation has occurred or not. But initially, as we have already mentioned, that Penicillium notatum, it produces penicillin in static condition, not in starved condition. And now we can see that the improved strain, the modified strain, can produce penicillin with a high yield in continuous start tank type reactor. And in the scale-up process, we can basically prove this. Let us discuss about the processes through which we can generally perform this mutation or mutant selection. So here we can say that there are 4 methods. Number 1, replicating method, resistance selection method, substrate utilization method, and carcinogenicity test. So these are the tests we can use for isolating mutant or selecting mutant. Basically, the first technique, that is replica plating technique, was coined by, was discovered by Lederberg in 1952. And this technique is used to detect auxotropic mutant that which differentiates between the mutant as well as the wild type strains on the basis of the ability to grow in absence of an amino acid. So the oxanographic method we have discussed in our first lecture. Now the second method, that is resistance selection method. In this method, we are using this method for isolating mutants, and generally the wild type cells are not resistant either to any antibiotic or to bacteriophages. Therefore, it is possible to grow other bacteria in the presence of those agents, either antibiotic or bacteriophage, and we have to check whether they are surviving or not. And it is very important, because one of the biggest hurdle we may have to bypass, that is contamination of bacteriophages inside the fermentation vessel. So if we can produce this resistant strain by using resistance selection method, then chances of contamination or proliferation of bacteriophages, if a single bacteriophage is present inside the fermentation vessel, it will not impact on the overall productivity of the bacterial strain. Now, the third method that we are going to discuss, it is the substrate utilization method. And this method basically we are using for selecting bacteria. We know that several bacteria they utilize primary carbon source for their growth. Now the cultures are plated onto medium containing an alternative carbon source that we have already discussed about the volatile carbon source during secondary screening. Sorry, during primary screening. So any colony that grows on this alternate carbon source, and they can utilize this alternate carbon source as a substrate. So through this, we can basically identify a potential mutant. And these mutant can be used if they produce certain bioactive compound. We can use these mutants industrially. The last one, that is carcinogenicity test to identify the environmental carcinogens that causes mutation and induce cancer in organisms. And it's based on detecting potential of carcinogens and testing the mutagenicity of the bacteria, mutagenicity in the bacteria. Next, we will discuss about random selection. So random mutagenesis and selection are also referred to as the classic approach of strain improvement procedure. Here, improved mutants are normally identified by screening a large number of population of mutated organisms. Why? Because the mutant phenotype may not be easy to recognize against a large background. And after inducing mutation in a culture, the survivors from the population are randomly picked and tested for their ability to produce the metabolites of interest. Now, in this approach of strain improvement, it involves repeated applications of three basic principles. The first one is mutagenesis of the population to induce genetic variability. The second one is random selection and screening from the surviving population of improved strains by small-scale fermentation model. And the last one is assaying of fermentation media. It could be broth, it could be agar, for developing products and scoring the improved microbial strains. So it must be emphasized that the action of mutagenic agent on DNA not only can cause genetic alteration, but can also induce cell death. And owing to irreversible damage to the DNA or formation of lethal mutations. Hence, after the mutagenic treatment, mutants are sought among the surviving population with the anticipation that each of the surviving cells harbors one or several mutations. And each time an improved strain is derived through mutation. And it is used again as the parent strain in the new cycle of mutation, as well as screening by fermentation media. So here we can say that the random selection method of random selection procedure of mutant selection is continued until a strain is derived that is statistically superior in performance to control the strain prior to the mutagenic treatment. Now we will discuss about genetic recombination. So in addition to manipulation of microorganism through mutation, the technique of genetic recombination can be uh, employed to get new strains containing novel combinations of mutations, and also to produce superior microbial strains. Generally, genetic recombination methods include those techniques that combine two DNA molecules having similar sequences, means that are homologous to each other. And through the spatial event of crossing over, they are reunited to give a new series of nucleotide sequences along the DNA that are stable, expressible genetic traits. So this mechanism of gene alteration and strain modification is known as genetic recombination. And this definition basically includes the techniques like protoplast fusion, transformation, and conjugation. Let us discuss what is protoplast fusion. Protoplast fusion is basically a widely used technique. Mainly it is widely used with yeast and molds. Most of these microorganisms uh, are asexual or uh, having a single mating type, which decreases the chances of random mutation that could lead to strain degeneration. So to carry out genetic studies with these microorganisms, protoplasts are prepared by growing the cells in an isotonic solution, while treating them with enzymes like beta galacturonidase or cellulose. So cellulose is the medium, and beta galacturonidase is an enzyme. Now, the protoplasts are then regenerated using osmotic stabilizers. One of the osmotic stabilizers used in protoplast fusion is sucrose. And if fusion occurs to form hybrids, desired recombinants are identified by means of selective plating techniques. A major advantage of this protoplast fusion technique is that protoplast of different microbial species can be fused, even if they are not closely linked taxonomically. Still, they can be fused. For example, we can discuss here that the protoplast of Penicillium roqueforti, which have been fused with Penicillium chrysogenum. Even yeast protoplasts can be fused with erythrocyte. Now here we will be giving one picture for this. There are three stages. In stage 1, you can see the myeloma cell and the antibody producing spleen cells. They are incubated in a spatial medium containing polyethylene glycol, PEG, which is basically, this PEG is basically functioning here as an inducer, which enhances fusion. And at stage 2, you can see that the myeloma spleen hybridoma cells that are selected out and cultured in close agar dishes. In the third stage, the specific antibody producing hybridoma is selected and propagated in culture vessels as well as within animal. Means both we are propagating this in vitro and in vivo, and also we are preparing monospecific monoclonal antibody. And this monoclonal antibody can be harvested through this. So in this way, by using this technique, you can prepare monoclonal antibodies. Now, next process is transformation. So transformation is the process involving the direct uptake of purified exogenously supplied DNA by a recipient cell, or it could be a protoplast. And when this uptake occurs, the donor DNA may either combine with the recipient DNA or exist independently within the recipient cell, which leads to changes in the amount and organization of the recipient microbial DNA, hopefully improving it with some of the characteristics coded by the donor DNA. So through transformation, transformation can be mediated by total genomic DNA or can be cloned sequences in plasmid or phage DNA. Essentially, the cultures to be transformed are cultivated in a specific physiological manner to develop the competency to make them readily accept the foreign DNA. Now, next process is conjugation. So this particular process introduces mutational changes in microorganisms through unidirectional transfer of genetic material from one strain to the other strain. And it is mediated by plasmid sex factors. It requires cell-to-cell contact and DNA replication. So this mode of genetic exchange can be achieved or can achieve transfer of chromosomal DNA or plasmid DNA. And several strains have been modified using this approach. Now we will be discussing on in vitro recombinant DNA technology. Now by using restriction endonuclease as well as a ligase, investigator can cut and splice DNA at specific sites. Now, some endonucleases have the ability to cut precisely and generate what are known as sticky end. And through a form of biological cut and paste processes, the lower part of the of one DNA is made to stick well onto the upper part of another DNA. And these DNA molecules are later ligated to make a hybrid molecule. And the ability to cut and paste of the DNA molecule is the basis of genetic engineering. And the useful aspect of this cut and paste biological process involves the use of plasmid, use of phage, and other small fragments of DNA, which we consider as vector. And all these entities are capable of carrying genetic material, as well as inserting it into the host microorganism as foreign DNA. And after entering inside the recipient cell, it will replicate and express within the host. So a wide array of techniques can now be employed to isolate, sequence, or synthesize, as well as modify or join fragments of DNA. And it is therefore possible to obtain nearly any combination of DNA sequence. But there are certain challenges. The challenges lie in designing sequence that will be functional as well as useful for the industry. Now we will discuss that the protocol to modify and improve microbial strains. Now, what are the steps we have to follow to modify and improve microbial strains? First, we have to isolate the desired gene, means the DNA fragment, from the donor cell. Then we have to identify an ideal vector. It could be a plasmid, it could be a phage. Then we have to cleave the vector, align the donor DNA with the vector, and insert the construct or insert the gene into the vector. After that, we can introduce the new plasmid into the host cell using the process that is known as transformation. And if you are using a viral vector, the process is known as infection. Then the last step is to select the new recombinant strain that express the desired traits. Now we will discuss about the improved strain performance. Through engineering optimization, major improvements in fermentation are no doubt attributed to superior strain created through mutation, through genetic alteration, or through genetic engineering. Further improvement in culture performance can also be achieved by giving a strain the optimum environmental as well as physical conditions. And during strain improvement process, it is important to keep in mind that the ultimate success also depends on optimizing the fermentation process, optimizing the fermenter design, and the use of either the batch or the fed batch or the continuous culture. The extent of shear force or the broth rheological properties as well as oxygen mass or heat transfer characteristics all attribute to improve the strain performance. It's not solely the genetic traits, but also the physical attributes. Fermenter design is equally an important critical parameter, and collectively they contribute to improve the strain performance. And the application of biochemical engineering principles can be used to design the fermenter and also to control the environmental parameters. That basically shift the kinetics of metabolic activities or metabolic pathways towards the desired product. And after the successful introduction of the improved strain into fermenter, production processes are validated and designed to run automatically to comparative and consistent operations. Typically, this is done in 3 stages. First one is known as is the qualitatively analyzing relationships among growth, substrate consumption, and production. The second is establishing mathematical formulations as well as kinetic equations of a model. The third one is estimation of parameters as well as simulation of the model on the basis of experimental data. And all these steps are very important and that are used in the scale-up process as well as modeling studies that emphasizes that how the product will be formed or how the fermenter will be operated. That's all for today.